Morning, everyone. I'm really sorry I couldn't make it yesterday, but I went to a very nice wedding, so I could tell you about it. It was very interesting. And I feel like because of all of the talks yesterday, I feel a bit like I'm going to be repeating things that you talked about, maybe with my own slant on them. So I'm hoping, and I've already heard just around the breakfast table I was sitting at, there was already a lot of discussion about definitions and issues like that. So I don't have a lot of slides, so I think we have plenty of time. So if you want to make a comment or you have a question, really feel free. Just want to point out, this is something I just discovered, Academic Commons at columbia.edu. Almost all of my papers are deposited there. I'm slowly working on it, and so just for FYI, here's the acknowledgments I had to show you. Really, most of this is theoretical, but Chris and really Jason Steffener, you'll see, played a very strong role in my thinking, especially about the imaging side of this. Okay, so I heard you have this discussion already about definitions, and I think really what we have to do if we're going to work in this field is get to the point where everybody knows what everybody else means. That means even if you don't want to accept someone else's definition, you at least know what they're talking about. And these are sort of fuzzy concepts in a lot of ways, so I think a lot of work needs to be done in just getting everyone to agree on what's what. So I just went through this process with this white paper, these PIAs from the Alzheimer's Association, their professional interest associations. For those of you not Alzheimer's related, you probably never heard of them. So we have one called Reserve and Resilience, and we actually put together this white paper. And the idea was very simple. It was about 30 people in the work group, and to come up with common definitions for reserve, brain reserve, brain maintenance, and cognitive reserve. We also treated the word resilience. I have to tell you, across, Rich was part of it. I don't know if anyone else in the room was part of it. Maybe Anya. Yeah. Can't see that far, but someone else raised their hand. I really can't. I'm sorry. I'd say there were, first we started with maybe 6 people. I wrote a draft. I thought, this will be fine. Major revisions through 5 or 6 rounds. And then we put it out to the work group. And again, huge numbers of revisions, quibbling, etc. And what we did is we tried to come up with definitions, potential measures, and research approaches. I think this is going to be an ongoing process. So I was just talking. I think I brought the PDF. There's some repository you could put it on, so everyone could take a look, especially the research methods, which is what we're talking about over this meeting. Very rudimentary, very basic guidance. You'll see what I have on here. But I think it's a start. Brain reserve, more neurobiological capital to lose. That's the definition. It's much longer in the white paper, but that's the idea of brain reserve to our point of view. I think that the person who used the term brain reserve first was probably Katzman. We were just talking before about how the Blessed Tomlinson and Roth paper said something like that as well. Brain maintenance, reduced development of primary age-related changes, age-related brain changes and pathologies. That's a relatively new concept, and that's not one that I really thought about when the whole cognitive reserve thing started, because we didn't understand that exposures could reshape the brain, but now it's very clear that that's the case. I guess the newest discussion is not more about age-related changes, but pathologies. Can we do things that will lower the risk of developing amyloid or plaque, etc.? And cognitive reserve. It's not usually my definition. Adaptability, flexibility, efficiency, capacity, compensation of functional brain processes. So this is the brain, the substrate, hardware, software. And those were, it's much longer than that. So those were the basic definitions that we came up with. If you look at the manuscript, you'll see they are really elaborated upon, I think in a very clear way. There were some really interesting discussions. The brain reserve versus cognitive reserve on two levels. So some people don't like that differentiation. The troublesome part of it is that we're not dualists. Cognition sits in the brain. Cognition, requires the brain. And so there's debates on that on two levels. So there's this paper coming out, I don't know if it's accepted or not, by Roberto Cabeza and a whole bunch of people who really were working on something called compensation. But they decided, well, cognition is in the brain, so let's just call this thing reserve and say it's sort of an early process. But these were people who really are not very schooled in the whole idea of what we're working on this weekend. They're thinking more about how the brain activates more when you challenge it. But there are also people that are very strongly involved in the cognitive reserve concept that are having trouble with this differentiation. My feeling about it right now is very simplistic. Let's take someone like Dave Bennett, right? He has people's brains, or the Rush. He has people's brains. They do the most amazing analyses up to proteomics of the synapses and everything. They still cannot suss out the cognitive networks. There's just no way they can do that. Maybe one day we'll be able to. So there really is a difference between what we can see of the brain substrate versus the cognitive networks that we can measure with things like resting bold or activation studies. So for me, that's the differentiation right now. It might not be completely physiologically accurate. I'm not a dualist, but the truth is that we can really, I think, differentiate between the brain substrate, the best we can measure it with imaging or whatever else, and the cognitive processes that we can do through cognitive experimental research or functional imaging. So that's where I come down on that. And then the term resilience, which is a slippery one. In our hands, we just said, you know what? People use resilience in so many ways. We're just going to use that as sort of an overlapping term for all of this. That's resilience. And we did not give resilience a privileged place in terms of a process. And that's where we were at. Brain maintenance, part of the deal that we talked about was brain maintenance, is that the process of brain maintenance determines the amount of brain reserve anyone has at any one period of time. So at any point in time, you have a certain amount of brain reserve that has been influenced by brain maintenance. And then we also offered some potential measures and research approaches. So it was a very interesting process. I think that process is going to go on, but it's really important. We need a common terminology. And especially as we start to invite our colleagues in the more basic sciences to help work with us, we really need to get these straight. So here's a paper that came out just last year. Dendritic spines provide cognitive resilience against Alzheimer's disease. It's a very nice paper. Maybe some of you saw it. They found that increased spine extent, they had these very intricate measures of spines. And with that, they could take people who had pathologic AD, Alzheimer's disease, who did or did not express Alzheimer's disease during their lifetime, and they could differentiate them based on this dendritic spine extent. And they said, these observations provide cellular evidence to support the hypothesis that dendritic spine plasticity is a mechanism of cognitive resilience that protects older individuals with AD pathology from developing dementia. So what is cognitive resilience? And I think that's where we have to just develop a common language. I'm not saying that's a bad term. I'm just saying let's try to use terms that everybody understands. What would I say that this increased spine extent is? I would say it's a form of brain reserve. Or if it's developed via certain exposures, it's brain maintenance in process. Does it support cognition better? Yes. But in and of itself, it's not cognitive reserve. But that's me. I feel like I really know it, but that's, yes.
I have a question.
Yeah.
So how would that be?
Hold on, Monica. Can we interrupt you?
Yeah, please.
I'm sorry. now in New York.
I was
I'm so
trying
rude,
to.
sorry.
I was trying to invite some. Yeah.
Okay, that's what I got from you. Yeah.
Yeah.
So thank you. So my name is Monica Rivera-Mint. I am from New York City now. I'm at Fordham University and Mount Sinai School of Medicine.
Why do you say now? How long have you been there?
16 years.
Okay, so you're trying to pretend like you just got there.
I'm originally from California, so I was carried in my heart.
Okay, go ahead. I'm sorry, Monica.
By the way, and thanks for giving me a hard time. And I thought I was into. So I have a question about this. So this sounds. I really want to read this paper. It's interesting, but can you help me understand how this would be different from just thinking about it like neural plasticity?
Well, it could be neural. So the real question is, how did this dendritic? They didn't look at plasticity. They just saw some people had a greater extent of these spines than others. How that occurred, we don't know. Were they born that way? But from my point of view, simplistically, here's a very interesting finding that gets down to the basic anatomic level and shows perhaps how more resilient cognition, more flexible cognition might be supported. But to me, it's a basic brain mechanism. Anyone else have a thought about that? Yes. Go to the microphone so you can be recorded for posterity.
Yes, so this is Paul Crane from Seattle, and thanks so much for coming.
Hey, Paul.
So a couple questions. The first one is, it sounds very much like it's technology driven. So I'm very much in sympathy with David Bennett. My office is 2 doors down from the neuropathologists in Seattle. And I look over their shoulder all the time. It's fascinating. And in my brief lifespan as a scientist, we've developed a whole new strain of TDP-43.
Right.
Where we didn't have it before.
Right.
And I'm also very aware that the pathologists only look about 12 places in the brain at the microscopic level.
Right.
So if we looked at 50 places in the brain, or 1,000 places in the brain, we would find more stuff.
Right.
And with TDP, we went back through brains that had already been fully characterized, and so you could parse out which bucket are we putting people into. And it sounds like if you can see it by the pathologist, then that's, or you don't see something that the pathologist would see, that's brain reserve. And if you do, like if it's, so if it's something that pathologists can see, it gets into the brain category. And if it's not, then it's the cognitive category. But the pathologist has new tools all the time.
Yeah.
New places to look all the time.
Yeah.
And is that really the distinction? Is it technology-driven current practice?
I hear what you're saying. So, I don't think it's just what the pathologist can see. I think we also have amazing structural imaging techniques, DTI, white matter, cortical thickness, et cetera, et cetera. So this is sort of the argument that, I had this discussion with Roberto. He's saying, well, we can't now, but eventually we will be able to. So let's say we could really get to the point where we could image the brain so well that we understood the complete brain substrate for how cognition is mediated and flexibility in cognition. I think at that point we would be at a totally different level. I feel that no matter what pathology does, we're not going to get to that point in the next 50 years.
But if we do, then it would all be brain.
If we do,
we
could call. Well, then I think we'd have to start to figure out what to do with that, because again, basically, it's hard for me to even imagine that. But basically, you took the same neuroscience 101 that I did. You have synaptic connections, and you have, so basically they'd have to understand these synaptic connections so well that they could determine exactly what these cognitive networks are.
Yep.
I don't think that's really going to be there. I just don't, they're trying to. So once that happened, I guess we could explain all this on a mechanistic level. And we would still sort of have to say, well, here's the substrate for these cognitive networks, and then we know the neuronal processes that propagate networks. So at that point, things would be a little more loosey-goosey. But right now, I don't think we're even close to that. And for a lot of reasons, I think it's useful to keep those two things. Let me just go back a slide. Can I do it? Yeah, I think right now it's where we're at. I think it's important to keep those two things separate because really the main question we're all asking is what moderates the relationship between the brain and how people behave cognitively and functionally. And we need that background to do it. So yeah, I admit that one day maybe this differentiation is going to be totally useless. But right now I feel it's very important. Age-related changes, Alzheimer's-related changes, what other brain changes that we can identify. And you're right, the more we know about the brain, the better it's going to be. Right now, we have this weak idea, especially, never mind Alzheimer's, at least there we have some heavy hitters. Cognitive aging, if we want to moderate against something, we have to figure out what we're moderating against. Is that the right word to say? To moderate on, moderate against. To moderate, just to moderate. And for normal aging, we're still struggling with what are the brain changes that seem to be involved in cognitive aging. So that's a long-winded answer to your question.
And it leads directly to my second question,
Yeah.
which is the brain maintenance bullet point. So the reduced development of something, reduced is such an interesting word. Is this a within-person phenomenon? Can you observe brain maintenance in one person? Or do you need some sort of comparator who does have the primary age-related things?
Right.
Can you observe brain maintenance in one person? Or is it all that you do is that you observe the absence of disease?
So,
Along my mind, I know something.
yeah. So, I mean, it's a very interesting question. I'd say where we're at right now, yeah, it's a differential deal. So, some people seem to have maintained their brains much better than others. Those people had XY genes, these life exposures, etc. I think when we try, a lot of people try, I'll show you some data later. We have a rough and ready measure of brain maintenance where here's what someone's brain volume typically looks like at that age. This person is worse than that or better than that. That's an N=1 with an implied comparator. But yeah, that's where we're at with that.
So I would say you can observe brain maintenance within a person longitudinally. It's hard to do cross-sectionally.
But how do you know that they're maintaining?
You can just see if the brain changes.
So you're saying if someone really does not show any volume loss and doesn't develop, huh?
You're still comparing that to a volume loss.
Yeah, I mean, I guess you could say if someone never develops Alzheimer's.
So if they're losing volume,
right?
You know they're not maintaining.
Maybe they're losing volume slower than someone else.
You don't know that they haven't dropped down from a level. But that wasn't my question.
Yeah.
Could you go to the slide that I think it's the study?
Yeah.
Yeah.
I thought this is a good topic for discussion, right? Because it gets right at the.
This is interesting.
Yeah.
This, having been around for a long time, I remember things, but I forget a lot of things. And this struck me because back, 25 years ago, Bob Terry and Bob Katzman were talking about synaptic density very much along these lines.
Right.
And they had empirical data.
Right.
And I believe it was synaptophysin that they were looking at. But, this strikes me as being very similar to what they were doing.
Well, there's a
big technological advance.
Well, this is not synaptic density anymore. This is some specific measure of the flexibility of some aspect of the dendritic spines.
Right.
So it's a little different, but it's the same sort of idea.
Yeah, and the point is that the technology, I think what the notion of Terry and Katzman always was intuitively very pleasing. It made a lot of sense.
Yeah.
The technology, it was really difficult to do that.
So
this in technology, we can really do things that they sort of knew 25 years ago were important.
I agree.
Now we're getting to the point where we've
I agree. So I'll tell you some more inside baseball about this white paper. So I submitted it because these PIAs are part of the Alzheimer's Association meeting. So I submitted it. I sent it to Alzheimer's and Dementia. That's the association's journal. Zavin, Kachaturian, and Ara. And I said, look, this is from a PIA. You got to publish it. They said, well, we're sending out for review. I said, you can send it out for review, but unless there's something major, I don't want to send it back to 30 people for revisions. I played a little bit of hardball. So they did two things. First, they said, well, for the people who are not into this, can you write just a little introduction about how this whole idea of reserve? So I wrote an introduction. Then they said, well, immediately Zavin, we want to write an editorial to introduce this. And the editorial pretty much says what you just said, that really this is all about neuroplasticity, and it really gets back to Katzman and Terry's synaptic density. It's the most wild editorial. I edited it enough that they put me on as a co-author, but and basically what they're doing in this editorial is inviting more papers on this topic, and especially basic science. Yeah, so that's the tack they took. Yes?
Hi, I'm Teresa. I'm from California now, but I was from New York before.
You're a New Yorker.
I'm
Yeah.
So I really like the way that you broke down those definitions in the previous slides of reserve maintenance and cognitive brain reserve. Anyways,
Yeah.
What I'm saying. So, but then, when you were reading this second bullet point on the third bullet point about,
This one?
No, right there.
Yeah.
So these observations provide cellular evidence. So that line there, I was like, oh, so according to your definition, that must be cognitive reserve. And then you said the two other ones. But this is what I'm wondering, like if AD pathology is present, but it prevents people with that pathology from getting the dementia, isn't that exactly what cognitive reserve is?
Well, I would say that their brain substrate, the idea of brain reserve is, what's the most simple idea of brain reserve? I mean, what Katzman said. They have more synapses to lose.
Right.
Or these people have synapses that support cognition better. That, to me, that's the brain substrate. It's the brain substrate. So it's a reserve still. It's not that it's not reserve. It's just not a cognitive process. It supports a cognitive process, but it's not a cognitive process. I mean, this is just me. I could be wrong, and you could disagree.
No, it just seems like, I guess from the way you defined it, it seemed like cognitive reserve was that gap between people with the pathology. They have the pathology, so they don't have the maintenance because they've developed that pathology.
No, but
they don't have the reserve either.
But even in the face of pathology, there still is brain reserve. And there still is cognitive reserve.
Okay.
Yeah.
All right, and
Yeah. I mean, I think these are the discussions. We have to get to the point where even if people disagree, they say, well, some people would call this brain reserve. Other people would call this, whatever he called it, cognitive resilience. And they pretty much mean the same phenomenon. If we could at least get to that point, it would be a big, because you guys are here because you're interested in this, but in the outside world, first of all, there's people who have never heard of cognitive reserve, and these words are used in all kinds of interesting ways. I see it because I review a lot of papers. Okay, so let me move on a little. So cognitive reserve, for a long time, we've relied on these proxies. What are these proxies? Basically, from my point of view, what happened was there were these original epidemiologic studies. They started with incidence of Alzheimer's disease, where people with higher IQ, education literacy, occupational attainment, engagement in leisure activities had reduced risks of developing dementia. And we said, oh, these life activities must in some way promote cognitive reserve. So we'll use these as proxies in our research. That's all they are. It's important to note that they're not direct measures of cognitive reserve. No matter what you do with them, they're proxies. If they behave like reserve, that's what we want to see. So, and I just wrote this because I don't know how many papers I've reviewed like this. Just showing that some task correlates with one of these proxies did not indicate that that task measures cognitive reserve. I mean, it sounds simplistic, but I get papers like that all the time. Oh, here's a digit symbol correlates with IQ. It must be a good measure of cognitive reserve. Yeah. Rather, it has to act like. Act like cognitive reserve. What does act like mean? I think the most hardcore definition moderates between brain and clinical outcome, cognition, function, etc. And and as you know, there's instruments that have been created that attempt to collect a lot of these proxies. And there's many more I didn't list. The Wilson scale, physical activity, etc., etc. Rich taught me this. 9 years ago. Maybe, did we talk about it at the meeting, or you thought about it after the meeting? This paper. Yeah, this was a paper.
Been here last night.
Huh? Yeah, you were talking about all this stuff? Yeah, so I got this online somewhere. You didn't make those figures, right?
I did.
You did? Those are yours? Yeah? Probably?
I stole them from somebody.
Whatever. So you guys know all about this already. So in a reflective model, that's what we're used to. You have 7 tests of attention, and they reflect the underlying construct of attention. I like that stuff. We're doing stuff that kind of stuff with imaging now, where we're looking for latent variables. The cognitive reserve is more like a formative model, where education, occupation, leisure activities all contribute in their own way to reserve. And I think that's a very important thing to measure, to think about. And what it gives me pause about is creating like a summary measure. Oh, my summary measure of reserve is a combination of education, IQ, and leisure activity, and I add them up. Or I did a factor analysis of all of these. I came out with one latent factor, because what you're doing if you treat it like this is you're chipping off what's different and what's unique, what each of these experiences contribute. And I'll get into that a little more. So that's a very important point. And so I think when we create summary measures, at least of the proxies, that could be dangerous. Here's a nice paper. This is pretty old already, 2003. Marcus Richards from the birth cohort in England. So Marcus knows these people, well, not Marcus, the cohort has been followed since right after World War II. So we know SES, father's occupation, when the baby's born, cognition at age 8, education by 26, occupation at 43 years. And this was just a path analysis that they did. It's the exact same path on both sides. They just published, in their figures, they just highlighted 2 separate sets of connections. Their point was an interesting one. Maybe it's just IQ, and people with higher IQ get a higher education, get a higher occupation. So their point is whatever endpoint you look at age 53, which is a little young for this, there's differing contributions of each of these, independent and unique contributions of each of these to the final where people are at later. I think that's really a very important thing to remember. The reason I put the slide here also is because what you're seeing is that these are the proxies we use. I think for every outcome, each of these is going to make its own unique, each of these life experiences, whatever you want to call them, exposures, is going to have a unique contribution, and it really might differ on the basis of what it is that you're measuring, what the outcome is that you're measuring, or what you're looking for in terms of an outcome. Simple point. And you see that for Nart, the relative contributions of these are different. He's just in the middle. I don't know if he published it yet. I just spoke with him the other day. He has a paper coming out with the newest best. They later worked in leisure. This is a life course study. So, this is the optimal way to play this game. They have these observations. And they're really beautifully in the sweet spot now, where these people are in danger of dementia. They're imaging them up the wazoo. So I'm a lot jealous. I sort of would like to go to London for a year or 2 and just play with those data. Maybe I'll do that. Okay. My friends know that I can't give a talk without showing this slide. It's the first paper I ever published on cognitive reserve, and I didn't even call it cognitive reserve in the paper, it seems. Someone just pointed that out to me. But basically what we did here, and Laura talked about this yesterday, is we controlled for clinical severity, and we had this measure of Alzheimer's pathology. What would we call this now? This is a Xenon scan. Neurodegeneration. We were way ahead of our times. It's a much better measure than plaques or tangles. It's neurodegeneration. It's a CBF measure that's sort of the final common path of whatever is underlying. And we found that the people with the highest education had more neurodegeneration than the people with lower education, given so they're walking around. These people are walking around with more pathology than these. And I just wanted to show, the reason I put it up is because I like to show the slide, but I'd show you how we did it. This is a long time ago. It was really Gene Alexander. I don't know who knows him from Arizona. He was our RA then. He did these analyses. So here we controlled for mini-mental, function, age of onset, duration, and we accounted for 19% of the variance at this detector. Then we added in education, and now we're accounting for 30% of the variance. So we increased the amount of variance that we're accounting for. So that's not moderation, right? No, that's not moderation. That's what we published. So we said, okay, so this was, and then I'll just show you, and then this is the Dave Bennett paper. So he had the brains. Okay, that's much better. So he had a summary of AD pathology, global cognitive function proximate to death, and he gets an interaction effect, education by AD pathology, and you can see the interaction. So let's say given a certain amount of pathology at death, the person with 15 years was much worse off than the person with 22 years cognitively. This is already a moderator, an interaction. So this was a big set of discussions when we were writing the white paper. Rich chimed in on it, a few other people. Is it enough, just to show this, that you're accounting for additional variance over what the pathology is? Or is it necessary to show moderation? And we dithered on that. Finally, we said, well, probably moderation is better, but sometimes all you can show is that you're accounting for more variance. I think that it sounds like a small question, but it's one that we sort of have to come to terms with when we do our research. Here's the most simple models. For the next couple of days, you're going to be working on all kinds of a lot of arrows and pointers and circles and whatever. But really, that's what we're all about. So we sort of have to wrap our heads around that. And I think, that sort of speaks to, I'm sure you saw this slide yesterday also, the residual models were pretty much what you're defining reserve as what's left after you account for pathology and maybe some demographics. So that's not a moderating question, right? That's a residual variance question. But yeah, so the residual, and this residual acts like reserve. You guys heard this yesterday. So we talked about that in this white paper too and thought about it, not anything so super. So I like the residual models. They're very attractive. It's sort of a quantitative measure of the effect of CR at the present time rather than just a proxy. Theoretically, it could change as reserve is depleted. Someone with Alzheimer's disease, if someone has Alzheimer's disease, I don't care how much reserve they have. As we know, they actually progress more rapidly, and it's all depleted. At some point, there's nothing left. So perhaps residuals could help with that. What do we always have to remember? Especially, we just don't have, and we talked about it, maybe eventually we will, we don't have all of the brain measures to really account for the variance in cognition. We don't understand all of the brain measures that account for the variance in cognition, especially in normal aging. In AD, you've got these heavy hitters, plaques and tangles, although a lot of people in neurodegeneration, although a lot of people seem to live very well with those. The residual always harbors a lot more than CR. And then this is something I learned when this article came out. Laura was postdoc with us, so she did it on the YCAP data. And cross-sectionally, it was a breeze. But once you start looking longitudinally, it gets pretty hairy because there's a lot of moving parts. So you get people whose white matter hyperintensities and volume remain the same, but for some reason they're doing better on our tests, and boom, their reserve goes up. There's just a lot of moving parts there to deal with. I think it's an amazing tool. You just have to be aware of what you're doing. For the kind of stuff that I'm interested in, where I want to understand how reserve is implemented, and because each reserve proxy seems to contribute to that implementation in different ways, I wouldn't use a residual. But there's other approaches and studies where the residual is a very good, convenient, and high-level measure. You just have to, in your mind, you just have to know what you're doing and why you're doing it. Any comment on that before I zoom on? Okay, good. All right. So I just want to show you, this is a cute study that Chris did with residuals, but I'm doing it to make another point that is one of Chris's hobby horses. I'll tell you the answer before I show you the data. Chris is always upset when if two things correlate with each other, that doesn't mean, if two things correlate with something, that doesn't mean they correlate with each other, right? And people make this mistake constantly. We get bad reviews because. So here's what Chris did. He said, okay, let's use this residual approach to quantify brain maintenance and cognitive reserve. So for brain maintenance, basically, this is a little sidebar for the statisticians. Usually what people will do is they'll regress age against volume and look at how people veer from this regression line. But Chris, if you look at his paper, has this very cool proof about how that regression line ends up being contaminated by age anyway. So what Chris did for this is he just sort of did a nearest neighbor approach. So here's so he picked a window and he took some person and he saw how they varied from that window in terms of so this guy, the mean for the window around this person is here and this person is above it, so he has more brain maintenance. That's how he did it instead of regressions. He's 45 years old, 1.3 of the mean of her age-related peers in this 10-year window. And so he did this for 3 modalities: cortical thickness, cortical volume, white matter tract integrity, and he averaged them to get a brain maintenance variable. And notice he's using multiple measures, which I think is cool. We could add in another 15 if we really wanted to. The cognitive reserve is pretty much the way that it's always done. You predict cognition from a bunch of variables. Here I use volume, cortical thickness, white matter tract integrity, and the residual is reserve. What he did that's nice is he did it for 4 different abilities: memory, fluid, reasoning, processing speed, or vocabulary. Those are Tim Salthaus's reference abilities. And then the 4 derived measures could be described by one underlying latent variable, and that's his estimate of cognitive reserve using a residual. And then this is sort of the take-home, is that here, so his cognitive reserve and brain maintenance measure, these are correlations, do not correlate at all. So there's no correlation between cognitive reserve and brain maintenance, but cognitive reserve correlates with IQ and education, and the brain maintenance correlates with IQ and education, but they don't correlate with each other. So the reason I'm talking about this is that when we think about these analyses that we're doing, each of these things like IQ and education and all these other exposures make unique contributions, each to the how intact the brain is, and to cognitive networks. Each one makes its own set of contributions, and so we sort of have to keep that in mind in these mega models that we create. Yeah, and so this is the figure that we had in our most recent grant on cognitive reserve, where we were attempting to describe what we're doing. So let me try to walk you through it. We actually published it because Oziama Okonkwo had some special editions. So anyway, so I think it's not the best in terms of picking away at it from a statistical point of view, but here's the basic idea. You've got these things, brain morphology and integrity. Pathology I threw in there, but you could keep it privileged and separate if you want. That's influenced by age, genetics, life experiences. It's hard to really determine that, as someone said, maybe Paul, in cross-sectional studies. And so we can measure this brain morphology with all of these different images: gray matter volume, cortical thickness, white matter hyperintensity burden, white matter tract integrity, amyloid and tau burden, et cetera, et cetera. And a lot of what we think, why we do that, is we think that those things can help us explain the effect of age on cognition, right? So do these things mediate between age and cognition? And we're really just in the infancy of understanding, using imaging, cognitive aging. Okay, and then if you have this morphology or you have age, your CR should moderate between morphology and cognition. So that's sort of where a lot of us are at. What is that CR? How do we define that CR? Can we put cognitive processes on it? Can we have social development, et cetera? What I've been sort of fascinated by, I think I just was seduced with functional imaging because it was just coming out when I was thinking about these things, is can we use functional imaging? So in our studies, we give people a working memory task, an executive function task, and we have this task-related activation. And so what we said is, okay, perhaps what happens is that these morphologic changes can be seen impact task-related activation, which in turn impacts cognition. In other words, these brain changes are changing or influencing cognition. And perhaps cognitive reserve moderates that. And I'll show you quickly some evidence of that. So that cognitive reserve allows people to do better in the face of these brain changes. Why? Because their brain activation networks are more efficient, higher capacity, more flexible, whatever, right? And so, honestly, this simple mediation from here to here to here, even without cognitive reserve, is not trivial to establish. Because most people who do functional activation just relate it to a task, and they don't really use it so much to predict cognition. That's just the way it is. It's hard to do. And so we can establish this, we can establish this, we can establish this moderation, we can establish this one, but it's hard to get the whole Megillah into one analysis. And then I have this other line over here, and I'll tell you why, is because what we found is that even if you account for task-related activation, there's still differences in performance as a function of cognitive reserve. So two people are activating the same way, different performance as a function of cognitive reserve. We feel that that's some part of the reserve process that's just not captured by our task. So that pushed us to think for non-task related activation, or what I call task invariant networks. Really, I mean, where I'm at is, can we, besides saying that this reserve is happening, can we start to characterize these thought processes? If I was smarter back then, now I'm a little smarter, I wouldn't have focused on activation. I would have said, let's use cognitive experimental techniques and try to characterize the cognitive processes that are associated with better reserve. Can we create measures? If you think it's flexibility, can we create measures of flexibility? But the idea is to sort of get into that black box of what this reserve is. We know it's variability in function. So I think, in this meeting, you're not going to be using fMRI data, but we have done things like this. I'll show you in a second with resting bold, which is in the ADNI data set. It's in the Whitecap data set. So there are ways to have entry into that game. This was the one. This was to the right of that, because we were the whole grant was to now do follow-up on everyone. So he said, yeah, now if we have change in brain morphology and integrity, now we can really talk about brain maintenance, and we could talk about the cognitive reserve's effect on change in related networks. So that's sort of I don't know if it's the best model, but that's sort of a guiding model that we use, and the buckets that we put what we measure. This is a very early paper by Jason Steffener. There he is over there. Jason did these models. Did you talk about this yesterday or something like it?
I left it for you.
You left this one for me. This is Jason. Age, brain, cognition, ABC, and he set up these moderated mediation models. So this model here would represent, he called them lifetime exposures because it's brain maintenance, it's not cognitive reserve. So this arrow suggests that there's lifetime exposures that allow people to maintain their brain. That's brain reserve. Here's lifetime exposures that allow people to do better in the face of brain changes. That's cognitive reserve. And it's a very nice paper. He systematically tested all of these models in every ROI that FreeSurfer puts. FreeSurfer spits out a set of regions of interest, and he tested all of these models, starting with this one, then a pure cognitive reserve model, a pure brain reserve model, or nothing. Where he found most of his results was in the cognitive reserve model, where these life exposures moderate between brain volume and cognition. All of these areas that are in yellow, that's what he found there. So it's very basic. I think now you're sitting there with brain data, cognitive exposure data. So on some level, that's what a lot of you are doing. Does that mean my time is up?
No.
Okay, good. What I want to say is, then Jason and I tried to understand, well, what is this moderation? So, so this is the graph, and I'm showing you this for a reason. Here's fluid reasoning, GF. Here's the right putamen. And these are really cool graphs because everything here has meaning. The crossbars are means. The extent of these things are the standard. Yeah, you can read about it in the paper. So here's all the old people, the gray. You can see that their volume is lower than the young people. You could just sort of see that. And then what the moderation that we saw was that in the old people was, look, Here's the people with the thickest is the highest reserve. The larger the putamen, the better they're doing, where the people with lower reserve did not benefit from this larger. Now, I just made that up because that's the way the curves are going, right? I mean, I've just interpreted these curves saying, oh, what must be happening is these people can benefit from a bigger putamen. These people can't take advantage of it. What a lot of people look for in moderation, and Sylvie Belleville was she's a very good researcher in this area. She would say that really the moderation that you need is the opposite, such that the people with higher reserve can tolerate lower brain and still maintain function. So should we be so strict, or can we be flexible? But I think it's important when you're looking at these moderations to try to understand exactly what's going on. So this was just a fun exercise. And then this neural implementation of cognitive reserve. Again, this is from a paper I wrote a long time ago. So I won't bore you with all the bullets, but I'll tell you what I said. This is something that I just want to stress because it interfaces with the stack model. I don't know if you people are familiar with the stack model, but if you're not, I'll explain it to you. So I said if you think about the neural implementation of cognitive reserve, this was really pretty bad. I did this, but the reviewer sort of pushed me into it, and later I was stuck with it. Neural means neural implementation, so I differentiated between neural reserve and neural compensation. Those are two different ways that cognitive reserve can be implemented. And basically I said, look, what's going on? Is cognitive reserve something that just emerges when the brain is challenged? No. Cognitive reserve has to do with all these exposures we have through our life and how our cognitive networks develop. And then when we're challenged by a stroke or we're challenged by age-related changes, if you have a better developed set of cognitive networks, you can, more efficient, more higher capacity, more flexible, you can cope better. It's a subtle point, but it's one that a lot of people miss. So the stack model pretty much said the same thing. The stack model tried to explain how that happens, that when you're younger, you have certain networks, and then they're built upon and built upon. But they said, well, that's not like cognitive reserve. That's just something to deal with Alzheimer's disease. But if we think about what cognitive reserve is, it's something that's building up. That means that if we assess young people the right way, we can look for the neural implementation of cognitive reserve, even though these people, don't have brain changes yet. So it's just that's just me. I mean, that's how I see it. And then compensation, I reserved a very special definition for compensation, is that the use of alternate networks not normally used as a response to the disruption of normal networks. Now, some people, Roberto and this whole crew, just had a meeting on compensation. No, they said compensation has 4 forms: efficiency, capacity, reorganization that is useful, and reorganization that maintains function. And those are all compensation. I felt that you have the normal brain, and the real compensation occurs in the face of some pathology that challenges the normal way to do something. That's what guides me in imaging, but it's worth thinking about even in the kinds of analyses that we do epidemiologically. So Laura was concerned that I was changing my definitions, right? But does it make sense now what I'm saying? Yeah. And let me see. I have like 7 more minutes. I don't have a lot to show you. So let me run you through this very fast, just as a very, very concrete example of what I'm talking about. It's been published like crazy. We give people this letter Sternberg. They see 1, 3, or 6 letters. After 7 seconds, they have to say yes or no. That was a letter that I just saw. During this 7-second period here, they're just retaining that in their working memory. And we can see that the time it takes to make that decision increases, this yes-no decision increases as a function of load. So we said, well, let's look at this retention period and see what this increasing load does to task-related activation. So using this multivariate method called MLM, multivariate linear modeling, on the SPM data, on the general linear models data, this first network was expressed by both young and old people. Unfortunately, greater is down. The old people expressed this network to a greater degree than the young. In other words, they were less efficient, I would call that. To do the job, they had to express it more. we found a second network. It's even hard to illustrate it, but it came out significant in this MLM, that on the average, these open squares are young. On the average, young people expressed it around 0, and the old people expressed. And what was happening there is that the older people who were doing worse on the task, they had a greater slope of our reaction time in response to this increase from 1 to 3 to 6. They were expressing it more. And so Eric Zarin was with us then. You can read the paper. It's sort of fun because he and I had a little debate. He said, this must be something like dedifferentiation, where as the brain gets worse, people do something different. I said, no, this could, because he said compensation can't be associated with worse performance. I said, no, compensation could be the normal way doesn't work. Someone else, they're doing it a different way. And it allows them to do it, but not as well. Like a cane is compensatory. You can walk, but you can't run. And if you read the paper, you'll see that although he was biting his tongue, he included that potential explanation in the paper because I was the PI. So that's about why. And then later on, Jason Steffener came. So Jason, who now is in Canada, so Jason did something really cool. He traced the primary network, and he found that there was one spot in the primary network that had atrophy. And the more there was atrophy in that primary network, the more people were expressing this compensatory network. So he's already showing that it is a compensatory process. So that was a whole series of papers. So that's neural compensation. So that's pretty much the story. I think that we think about these ideas of what is compensation, what is efficiency, what is capacity. What I would say is, like for most of what you do this week, you're probably not going to get to that in terms of imaging, but you might be able to suss out some of that with cognitive measures. And it's worth thinking about. And then this idea of a task invariant network, the idea is that, well, if cognitive reserve allows people to escape Alzheimer's disease, it's not a Leonard Sternberg phenomenon. It's a global phenomenon. Can we identify a task invariant network? One that everybody invokes, and those who invoke it more do better. Simple as that. And so we did it. I mean, it was a long, crazy process. This is Chris. Essentially what Chris did is he took people 20 to 80. We had 12 different activation tasks. He sussed out a network, a covariance pattern that was expressed in all 12 tasks, whose expression correlated with IQ. Here's the things we wanted. Common across 12 tasks, expression correlated with IQ, whose expression moderates the relationship between cortical thickness and cognition, and whose expression in other tasks correlate with that proxy. And we did all of that. So the idea that there are some generic processes that underlie reserve, it's a theoretical issue, but I think it's sort of useful. What's sort of cool is if I don't think we really have the cognitive reserve network, and I'll show you why in 1 second. But if so, it would be, here just to show you. Here it is moderating between thickness and GF. And here it is, we forward applied this network to a totally different task, and expression of that network and that task correlates with IQ. So these are, these are more cognitive, for me, it's sort of conceptual stuff than us saying, well, this is the real network. But let's say it was. Then you could measure people's cognitive reserve, at least this aspect of it, out of an fMRI scan. You could, if you had an intervention to help people increase their reserve, you could look at the pre-post fMRI scan. It would be sort of cool. And to make life easier, we then did the same thing with resting bold. And we found I'll just go to the very end. We found a resting bold network that correlated with IQ, another resting bold network that correlated with education. And both of those we found that they moderated between brain and cognition. But I have to say that when you talk about moderating between brain and cognition, you have to figure out, well, what measure of brain you're going to use. What we're learning is that there's individual combinations of these brain markers that you're going to have to work with from YCAP and from ADNI, I guess from the other study as well. It's not like every cognitive domain is going to be equally affected by volume. Some are going to be more affected by volume, some more by white matter integrity, et cetera. You have to figure that out. I'll just go to the punchline. What we did then is we threw both of these into the mix. So we have now a education pattern and an art pattern. And we show that in specific cases, both of them moderate at the same time. So each are contributing separately, these two networks. Even though they correlate very well with each other, they contribute separately. So I'll stop there. So I don't think, it would have been cool, but the way that we do you have those resting bold networks, both in ADNI and I know in YCAP we have them, but the way we did it, it needs specific treatment before you could get these connectivity matrices. But if you wanted to, we could probably do it fast. Concept of reserve is promising because it provides resilience against yet untreatable diseases. Right? I think we all agree on that. And quantifying it is complex. That's the point I'm trying to make. I think the main point I'm trying to make is you really have to be, you can't get carried away with a lot of circles and arrows. You have to be very, very, very thoughtful about what it is that you're trying to measure and how you're doing it. Life experiences that appear to impart reserve has them used as proxy measures but don't directly measure CR. Residual measures have promise but rely on the assumption that, well, this is not really true, that all variance that is not accounted for by demographic or brain measures is CR. Functional resting bold might be useful. Here's what I'd say. At this point, care must be taken to use the potential reserve measures in the appropriate reserve context. And I think the sine qua non is moderating between brain changes and clinical changes, although we can't always get that level. Accepted, consistent definitions would be extremely helpful. Consensus on research paradigms, just so people understand and say, yeah, this is a good paradigm to use. That's why I'm really hopeful that I can distribute this white paper to you, because I'd love people to say, that was like baby steps that you described in these research paradigms. We can really goose that up. Because as you probably know, in one way or another, I don't know who's going to lead it, the NIA will fund a 3-year process to refine these once again. So we could really start on that. And common accepted test beds would be of great use. For example, we need accepted data sets in which to test whether a network or any other variable moderates between brain and behavior. Instead of everybody doing their own study, if we say, oh, here's a set of data for this cognitive process, these brain measures, you have some measure that you think is a reserve measure, bring it to this test bed. For imaging, particularly so, but even for life exposures or combinations of them as well. I think and forward application of findings to test data sets. Can we replicate? Can we have accepted places where everyone will say, well, wow, if you can show this and this and this and this data set, that really is a very strong finding. It's exciting to see this area of research expand and become more methodologically and analytically sophisticated. This meeting is really sort of the height of that kind of thing. Influencing reserve may delay or reverse the effects of aging or brain pathology. That's why we all get the grants, because that's our real hope is to do that. Careful, focused research will help us maintain the chance for successful intervention. So I think if we keep our research approaches thoughtful, keep our definitions clear, understand what our methodology is suggesting about how this works, it just will focus us a lot better. Okay, thanks. Okay, I'm going to go home now. So do we have time for questions? Or yeah. Okay. Uh-oh. Yeah.
Thanks a lot. That's perfect. I have a few comments.
Sure.
First of all, it seems to me that if we base our definitions on distinguishing between structure and function, every time the technology for measuring structure improves, what goes in reserve, cognitive versus brain, is going to have to change. So definitions based on structure versus function seem like a lost cause. To me, as basically a public health person, the distinction that matters, which gets really conflated with terms like proxy, is whether this is something we know how to modify. So education is particularly interesting because we know how to modify it. And there's a really big difference between a proxy measure that is influenced by a feature of brain that we don't necessarily know how to modify, like IQ, versus something like education, which we think influences brain, And we do know how to modify, and we could therefore use as an opportunity to improve cognitive outcomes for people. This relates to a larger point that is really like a big picture question about to what extent we care whether things like education are influencing cognitive reserve or just influencing cognitive outcomes. When we emphasize this idea that cognitive reserve is a variable that modifies the link between brain and cognition, it's important to recognize that what is an effect modifier is completely arbitrary, and you can change whether one variable modifies the other by simply transforming the dependent variable or transforming one of those two variables. So something that doesn't modify in a linear model, if you log transform, you can make it a modifier. Something that doesn't modify when they're both continuous, if you dichotomize one of them, you can make it a modifier. So this idea that we're going to say if it's just a predictor, it's not necessarily an indicator of cognitive reserve, but if it's an effect modifier, then it's cognitive reserve. That really is going to be very noisy depending on how you transform your variables, both the predictors and the outcomes.
I'm going to have a hard time memorizing all this, but go ahead, Marie.
I think that's my big...
Okay, so let's start with that, and then if you want to follow up. So yeah, I mean, look, I don't think we're in a hopeless situation. We're getting better and better at our brain measures. Imaging is getting much more sophisticated. As we discussed, path models and measures are getting better and better. I think we have to use the most up-to-date data possible. I think at some point it's not going to make a huge difference. You say, well, it's hopeless because it's changing all the time. Life changes all the time, or knowledge changes, but there's certain exposures that seem to be holding up. A. B, yeah, I mean, are there exposures that are part reserve that are modifiable and some that are not? Yes. Should we focus on only the modifiable ones? Well, that depends on your perspective in life. If you want to understand how reserve works in general, you might want to focus on all of them. If you want to focus on things that are modifiable, I think that's fantastic. Is education modifiable, late life leisure activities, etc. So to me, that's not a big issue. I think it's just a matter of our predilection for what we're interested in and understanding.
Distinguishing really matters for how we interpret the statistical associations, right? So distinguishing between things that are consequences versus things that are causes really changes how we interpret statistical associations. That's why, and certainly it matters for public health reasons, but it also matters for interpreting.
Yeah.
That's why I think it's important to be clear when we're using proxy if we mean something that's causing some feature of your brain or cognition, or something that is a consequence of a feature of your brain or cognition. Does that distinction? And I don't mean to say it's hopeless. I mean this particular way of...
So let's be concrete, like IQ versus education.
Yes.
Yeah, I know. I think it's important to understand what's going on and which might be genetically determined to a great degree. Although, it's pretty clear that IQ is genetically determined, but there's still a large amount of variance is probably can be influenced by life exposure. So and then I think the definitional part. So, I think a lot of people got hung up in normal aging. Let's say, especially the brain maintenance guys, Nyberg, and who's the other guy that I forget his name right now. I just saw him speak, and I told a bunch of people about it. So they're very hung up on the idea that, look, in most longitudinal studies of cognition with education, you're really getting an intercept effect, not a slope effect, right? He even wanted to hang differential incidence of AD on just that difference without remembering that there's Alzheimer's pathology that intervenes as well. So yeah, I mean, some of these differences are there. Is that cognitive reserve? The way I've defined it, just an intercept difference? Probably not. Does that mean it's not important? It's a pre-existing difference? No, it's there. But that's just the way that reserve was defined as moderating. Yeah.
Thanks. That was great. I just love to catch up with all the cool things you've been up to. And I really appreciate your model having multiple cognitive domains in it. I think that's really important in the concept of reserve. And the question I had was, I'm sort of perseverating on the Sternberg task.
Right.
And thinking also about Roberta Cabeza's work with non-selective over recruitment and just more, I guess you could say, targeted or selected or compensatory over
Right.
recruitment.
Right.
And his multiple different networks that he's been grinding for quite a while now.
Right.
And the network that you bring up as sort of as a cognitive reserve network is something that's multimodal. It is correlated with brain pathology in the end, and moderated in a expected fashion.
Right.
And I was wondering if I could press you for a moment on distinguishing it from, let's say, how it would play into this over-recruitment that you see so in all of the cognitive tasks
Right.
that goes on. And then also just to comment, the Sternberg task is really an attentional task. And so many of Roberto Cabezas is memory-driven, semantic, and there's sort of different levels to this onion.
Right.
Automatic processes versus
Right.
More effortful processes. And so the question is the network that you're recovering, is it an automatic network? Is it effortful? Is it,
Yeah.
what is it exactly?
Yeah.
As I sort of lay out this background.
Yeah.
Because I think
Yeah.
when you get down to intervention, are we going to intervene on effortful or automatic processes?
Yeah, no, I think that I have to say that I've been doing imaging to try to understand reserve better for a long time, and it's very frustrating because we're dealing with a lot of what everybody agrees on are very, very simplistic concepts. We've known for a long time that if you make a task harder, people activate more. We've known for a long time that older people activate more than younger. Let's just say this. Let's say that you have a task and you can do it two different ways. And someone who has the choice of doing it both ways will end up doing the task better than someone who just does it one way. The truth is that it's very rare to see an fMRI study that can capture that. Because you have to know too much. You really have to know exactly the activation signature of those two ways. And it sounds easy, but it's not. Yeah, so the whole question of what compensation is, what overactivation is, it's something that, so from what the reason that I got started with the letter Sternberg was because I was very hung up on difficulty. And I said, because what was happening originally, if you just go back to like Cheryl Grady's earliest paper, she was like the pioneer of this. She said, look at this, I'm giving people this facial recognition task, and the old people are activating the dorsolateral prefrontal cortex and the young people are not. That must be compensation, right? And then 2 years later, she said, wow, if I degrade these faces, the young people activate that area too. And this, I'm talking ancient history of me thinking about this. So I said to myself, wait a second, this is just a difficulty effect. This is not a compensation effect. If you make the task harder, they reveal more of this network. So I got very hung up on controlling difficulty, modulating difficulty as a window. And then there's this other fantasy behind it, that challenging the brain with difficulty is sort of like what happens when the brain is challenged by pathology, which is really not. So these are assumptions. This is sort of the path that I took. I'm getting more interested in sort of these more generic networks, although I think these task-related networks still have a very strong role to play in understanding basic mechanisms. Yeah, so I hope that answers your question. Yeah, I mean, it's very, very difficult to pull these things apart. But I do think that this, and I've showed you one end of it. The other side of it is what Patty calls the crunch part, the capacity part. You can see differences as a function of aging and differences as a function of reserve. So I think there's something to be said about them, but they're not the be-all and end-all. And what it really comes down to, and that's why we really want to get down to our basic science colleagues, is I guess I'm sort of a nihilist in terms of the networks and how well we understand these cognitive networks. But what I've really been spending a lot of time now on is like defining as best we can the cognitive networks that underlie Tim's four reasoning. And then I feel once we get those nailed down a little better, perhaps it'll be helpful. So yeah, these are very...
I was asking about the Sternberg task because that seems really specific.
No, I started with that just because it was an easy way to manipulate difficulty. So now what we're doing, just to tell you, is I have 12 tasks, 3 each from 4 abilities. We're getting a latent network for each of those 4 abilities. We've already identified them, and we're seeing how they relate to cognition along with all of the other brain measures that we have. And then, as Maria said, wait a second, now everything's going to change. You got another 17 measures to add in. But I think the more specificity you have in what really contributes to performance, the more you can understand what contributes to the individual differences. And if it's something that you can help people with, maybe make people better too. Okay, I'm here all week.